First of all, I thought that I will start a little bit of who I am and where I come from. So my background is in, let's see if I can get this working. Okay, good. So my background is in pharmaceutical sciences and biochemistry. And for the last a bit over 10 years, I've been working in the proteomics unit here in Helsinki. What we actually do is we are kind of a double-sided story. So we are operating as a proteomics unit where I'm in charge of the daily operations, but then we also have a research lab. And as the proteomics unit, we are aiming to really kind of serve the whole community, not only in Finland, but we have global customers from all over the world. And what we include in our analysis is basically global proteomics. This can be whatever type of sample the customer has. Most often it's a patient sample, eukaryotic tissues, cell lines, plants. We also do phosphoproteomics, interactomics, structural proteomics, and we are also moving towards the single cell proteomics. The main things that what we and our customers want to identify is basically changes in the proteomes in health and disease. And then, of course, identify also the cellular signaling pathways and biological processes that have been altered. Our scientific leader is Dr. Markku Varjosalo, and basically it's me and 3 other people who are involved in the daily operation of the proteomics core unit. What we try to do is we really try to do what Eric was also telling, that we work with the customers and for the customers. And first, we really try to find out what they actually want, to really dig into the biological question, the scientific question, what they want to answer. And as my colleague calls this is basically what Eric said is the pre-experimental conditions. So this is basically the quality control step -1. So we really need to think about what would be reasonable design for the study. Do we have enough controls? Do we have enough study groups? Does it really make sense to do the study as the customer wants to do it? And this is actually something that takes a lot of our time to act as consultants. But it's also very interesting. So basically, once we know what the customer wants to do and they know what they are supposed to do, so this is what we call the initiation phase. And while they are doing the studies, then we are basically making sure that we are ready for performing the analysis when the samples are here. At the same time, of course, our responsibility is to keep the instrumentation running 24/7. So we evaluate the instrument performance, we do preventive maintenance, we do checks, and basically, as you can think of, there are multiple of monitoring steps that we are doing on a daily and weekly basis. There are also then things like strategy and vision that come into the play, because we need to think where we want to be in the next 1 year, 2 year, or 5 years. What I would like to also share with you is our national survey from the users between the years 21 to 23. What I like about this is that proteomics was identified as the second most important service. We only lost to biological images, where you can think that the user amounts are also a lot bigger, so the N number to answer these questions was a lot bigger. What is cool is that we were kind of recognized as national #1 unit, saying that we have the state-of-the-art facilities and we are outstanding, very friendly, and really providing superior service with excellent quality, and we provide valuable input to the customer projects. I think that one of the major reasons of this very positive feedback was the instrumentation and the methods that we are using. And back in 2021, we had a change in our equipments. And this is when we got the first timsTOF Pro installed together with Evosep 1 and NanoElute. For the first half a year or so, we spent optimizing the methods and technologies with the NanoElute. And since August 21, we have been really operating with Evosep 1, I would say, roughly 90% of the time, together with the timsTOF Pro 2. We mainly use the 60 samples per day method. Sometimes we also use 30 samples per day. And with the 60 samples per day, we are mainly using the analytical column, the same as Eric was saying, the 8 centimeter performance. Since August 21, we have been doing roughly 20,000 runs with the Evosep. And basically the downtime during all of this runs and whatever has been less than 10 hours. Our weekly routine is basically consisting of running diagnostics with the Evosep, maybe not every week, but every second week to kind of follow up what's going on there on the LC. And then as a quality control sample, we are acquiring 50 nanograms of commercially available HeLa digest. And this helps us to really get through of what we expect from the LC and what we expect from the mass spec. Basically, during this couple of years running Evosep 24/7, we have had, I would say, 3 problems. So we had once a degasser problem. This was solved really, really quickly. Then we had a rotor seal, and once we had a leaking loop. What I really like about Evosep 1 and the EvoTips is the ease of use. It's really lovely plug and play and low maintenance. Coming from the world of different kinds of nano LCs, I would think that this is the LC that I've spent by far the least amount of time just problem solving and trying to think what's going on. Also, the EvoTips are really good for our workflows because even with little training, anyone can load the EvoTips. As we are a lab of roughly a bit over 10 people, then we cannot be serving all the people in the lab because we do have the customer projects. Also, because we are using the EvoTips, there is no need for separate trap columns. And this was something that we previously always used. So we had a 2-column setup. Also, as Eric said, the runs are really reproducible and they provide high quality data. And when we think about like 5 to 10 years back, when we were thinking of run times of 60 to 120 to 240 minutes, if we are using now the 60 samples a day, we are thinking about 21-minute gradient. So that's really a lot faster. One thing that both me and my colleagues are absolutely in love with is the interactive problem solving of the Evosep. So basically, if you have a problem with your Evosep 1, you can just go to the Evosep website and you can find a perfectly beautiful flow chart that guides you through the problem solving, and even down to the fact that, okay, this is where you have leak or problem. These are very detailed pictures of what you should do next. So that's absolutely something to recommend. What we have also noticed is that the EvoTips are not perfect for every single sample type. So that's why we also have the regular nano LC. And sometimes you would like to have maybe a 5-minute high organic gradient to just rinse out your system. But with the Evosep, you have the present gradients. That's also kind of the pro, because then you know that this gradient will work for my method. I guess that in the near future, we will have also a protocol for tip loading with Biomech i5. I hope this is coming, because this is what we are really looking forward to. All right, then let's go a little bit of what we have been getting out with the system. So let's go a little bit to the proteomics of NF-kappa B1. So Finland has a geographically isolated population, and the population is based on a small number of historical settlers. Although there is partly Scandinavian genetic influence and inheritance, there was geographical isolation of populations within Finland. This further led to genetically isolated populations, leading to special Finnish disease heritage. Thus, our population is rich in genetic variants that are not found elsewhere. Fortunately, there are high-quality genetic databases that enable exceptional possibilities to explore genetic questions. One of the specific features of the Finnish population is the high number of autoinflammatory conditions. You can still see that there are areas within Finland where immunodeficiencies are more common and with a common genetic background. And one class of these geographically enriched immunodeficiencies are the NF-kappa B dysregulation patients. I won't go very deep into the NF-kappa B pathway. I'll just say that NF-kappa B is the nuclear factor kappa light chain enhancer of activated B cells, and it's a key regulator of immune responses. and mutations in several of the NF-kappa B pathway genes cause immunodeficiencies. What we have been involved with is the multiple of NF-kappa B variant cases. Here on the slide, you can see the variants that have been identified in the Finnish population, in our Finnish kind of hospitals. The doctors are basically seeing the patients, and then they do the sequencing because they know that these are rare patients with very specific clinical laboratory parameters. And then they usually then find out that, hey, there is something that is usually a heterozygous NF-kappa B variant as the most likely cause of the disease. Usually, these are then the immunodeficiency and autoinflammatory patients. The symptoms vary, and the symptoms vary greatly in the carriers of the different mutations. How we then tackle the problem is via the interactomics approaches. So basically what we use is so-called MACTAC approach, multiple approaches combined. And the beauty of this approach is that basically we can have just a single cell line where we have expressed our protein of interest. And then we use one single cell line for both affinity purification to get the stable protein complexes. And then we use it also for bio-ID approach for proximity labeling MS to get the transient or proximal interactions. And this workflow is basically scalable. So it's kind of also something that we can then put together as a pre-step in the Evosep. While we were generating the bio-ID data, we also made a reference data set. And this is basically to study the subcellular localization of whatever protein we want to study. And basically, when we have the reference data set, we can see that where the neighbors of my interesting protein are, this is then where it's most likely located in the cell. And this approach is then termed MS microscopy. The resolution is somewhat similar to immunostaining, where the labeling radius is roughly 10 nanometers. Then when we think about what we actually saw with our constructs and our patient variants is altered protein-protein interactions. So basically on this slide, you can see that, for example, the arginine 157 stop only had very few interactions. And this most likely is due to the fact that there were only very few peptides also detected. And there are really varied effects from each of these constructs. Then when we do the bait-bait clustering of the APMS data, we can see that the point mutations are more heterogeneous in their molecular interactions, and the truncations then cluster better based on their location in the NF-kappa B1. When we look about the molecular context of the mutants with MS microscopy approach, we can see that there are really differences where these variant proteins are located in the cell. So basically, we know that there must be differences in their function, in their doings in the cell and in the patients. So to conclude, our studies significantly expanded the associations of NF-kappa B1 variants to immune system dysfunction. And the greatest thing is that we were actually able to deepen the understanding of NF-kappa B linked disease biology and improve patients' quality of life. So for example, the arginine 157 stop mutation carriers that at the time of the study were elderly brothers suffered from life-threatening sterile, so bacterial negative skin and tissue inflammation that they got even after minor trauma or normal elective surgeries. And after we understood better the disease biology, what was going on in these variant carriers, it was possible to treat these brothers with anakinra, so interleukin-1 receptor antagonist, and it really helped to heal the necrotizing fasciitis, which is a life-threatening condition. At the moment, there are constantly NF-kappa B variants spotted around the globe all the time. And we have analyzed more than 200 patients carrying more than 50 unique NF-kappa B variants. What we have noticed is that the phenotypes vary greatly from variant to variant and even within a variant. And most likely, as we have already published also with, for example, Sting, the underlying disease is kind of modified by other genes and other proteins and SNPs in those. I guess the great question is, will we ever fully understand the biology of NF-κB1? And I think that actually the challenge is not to find the variants, but really to understand them and their proteomics and high throughput proteomics, the systems biology approach, and then Evoseps, and then basically automated sample processing will be the key. I would like to thank every member in our lab, also our collaborators, and then also thank you for giving me the opportunity to present our story.